We've finished building our cell, filling it with cytosol and all of the organelles it needs to function. We can think of this as putting all of the furniture into a house. But what's holding the walls up or keeping the furniture from moving around? In a house, the framing and support beams keep the walls from falling in, and the floors and walls separate and anchor the furniture. Cells have a similar support structure called the cytoskeleton. The cytoskeleton is a three-dimensional network of microtubules and filaments. It provides internal support for the cells, anchors internal cell structures, and participates in cell movement and division. The cytoskeleton contains three primary types of fibers: microtubules, microfilaments, and intermediate filaments. Microtubules are cylindrical tubes 20 to 25 nanometers in diameter. Two proteins, alpha-tubulin and beta-tubulin, combine to form a tubulin dimer. Dimers come together in a spiral arrangement to make a long, hollow cylinder, a microtubule. Microtubules act as a scaffold to determine cell shape and provide a set of tracks for cell organelles and vesicles to move on. Although the microtubules might look like a jumbled mess, there really is some organization to them. Microtubules are produced near the nucleus in an area of the cell called the centrosome. A pair of small organelles called centrioles is found within the centrosome. These are made of a ring of 9 groups of 3 microtubules each. Centrosomes and centrioles both play a role in cell division, as we'll see when we study mitosis. Microfilaments are another component of the cytoskeleton. They're fine, thread-like protein fibers 3 to 6 nanometers in diameter. Microfilaments are composed mainly of a protein called actin. Microfilaments also play an important role in muscle contraction. Intermediate filaments are the third component of the cytoskeleton. Intermediate filaments are about 10 nanometers in diameter. They're made of twisted strands of proteins related to alpha-keratin, the main protein in hair and fingernails. Intermediate filaments have a lot of tensile strength that helps stabilize a cell structure. Like the rest of the cell, the nucleus also needs support structures. A network of protein filaments called the nuclear lamina is just inside the inner nuclear membrane. It's attached to both the membrane and to chromatin inside the nucleus and helps maintain the nuclear shape. Some of the proteins involved in structural support within a cell are also used in cell movement. Cilia and flagella are projections from the cell. They're designed either to move the cell itself or to move substances over or around the cell. Made up of microtubules, cilia and flagella both have the same internal structure. The major differences are their length and abundance. A flagellum is much longer than a cilium, and cilia are more numerous than flagella. Although cilia are very short, there can be hundreds or thousands of them in a single cell. Not all cell movement is due to external structures. Some cells have internal microfilaments that let it extend and contract in order to crawl along a surface. These extensions are called pseudopodia, which literally means false feet. Intracellular structures are also responsible for some types of movement within a cell. In large plant cells, as well as certain other cells, a layer of parallel actin filaments promotes the circular flow of cytoplasm, called cytoplasmic streaming. This motion helps speed up the distribution of nutrients and other small molecules throughout the cell.